Can you switch it on?
I was told we should move on. So the next talk is going to be delivered by Eszter Hegyi from University of Pécs. She's actually talking about the favorite topic of myself and Miklós, controversial genetic.
Our love story. Thank you, Andrea, for the nice introduction. And first of all, thank you, Peter, and thank you, Miklós, for the opportunity to present our data here. And I must say thank you, Jonas, for mentioning the CPA1 mice in your talk, because Miklós never mentioned that one. So at least you are fond of that mouse strain. So chronic pancreatitis is a multigenic disorder, and patients may carry even more than one genetic variant that alter genetic risk. And the majority of these variants affect the so-called trypsin-dependent pathological pathway of genetic risk in chronic pancreatitis. And in this pathway, premature trypsinogen activation plays the central role. However, some PRSS1 variants and also CPA1 mutations in the hybrid 1 allele of carboxyl ester lipase cause protein misfolding and consequent endoplasmic reticulum stress. And the ductal pathway comprises of genes that encodes channels which are predominantly expressed in pancreatic ductal epithelium, such as the CFTR, the Claudin-2, or the TRPV6. And in my talk today, I will focus on the two genes, the calcium sensing receptor and the hybrid 1 allele of the carboxyl ester lipase, and I will talk about their role in the development of chronic pancreatitis. So the calcium sensing receptor plays a pivotal role in bone and mineral metabolism, and it regulates parathyroid hormone secretion, urinary calcium excretion, skeletal development, and also lactation. And the role of a calcium sensing receptor in these calciuric processes is highlighted by gain of function and also loss of function mutation that causes familial hypocalciuric hypercalcemia or autosomal dominant hypercalcemia, respectively. The calcium sensing receptor itself is expressed in the pancreas, and actually Professor Gabor Varga and his group found that in the human pancreas, in the human pancreatic cells, this receptor is expressed. And probably it regulates the calcium concentrations in the tubes and may increase the ductal fluid secretion and may help to prevent stone formation in pancreatic ducts. So these variants may have actually potential role in the development of pancreatitis. And the first study that investigated calcium sensing receptors in potential chronic pancreatitis genes comes from 2003, in which Felder-Bauer identified a novel mutation of the calcium sensing receptor that is associated with chronic pancreatitis in a family with a heterozygous SPINK1 mutation. And subsequently, many groups identified patients with calcium sensing receptor variants and SPINK1 and 34S mutations. So what all of these studies suggested were that the calcium sensing receptor mutations might contribute to chronic pancreatitis risk, particularly in carriers of pathogenic SPINK1 variants. However, all of these studies had a major limitation, namely that there were case series or case reports. So they investigated a very few patients, and these were not case control studies. And the first study on a large cohort come from Professor David Whitcomb groups, and they identified a variant, the PR990G variant, which increases chronic pancreatic risk, chronic pancreatitis risk about twofold. And this was even more substantial in alcoholic chronic pancreatitis. And the French group tried to replicate these findings. However, they could not find the association in R990G variant Instead of this common polymorphism, they find an association with another common polymorphism, the A986S, namely that this variant in the homozygous state increases chronic pancreatitis risk more than threefold. So if we have to summarize the findings so far, they were very contradictory. So our aim was to explore the role of these two polymorphisms in chronic pancreatitis, and we had the Hungarian National Pancreatic Registry, and we included patients from that registry. We included 337 chronic pancreatitis patients and more than 800 controls, and we analyzed their exon 7 and especially these two polymorphisms. And what we found was that none of these two common polymorphisms associate with chronic pancreatitis. We did allelic distribution, and also we looked on the genotype distribution of both of these variants. And as you can see, there are very low odds ratios. There was no significant association of these common polymorphisms and chronic pancreatitis. And we also did a subgroup analysis, and we analyzed these two variants in the non-alcoholic chronic pancreatitis group and also in the alcoholic chronic pancreatitis group. But again, the findings were similar, so we did not confirm any association between these variants and chronic pancreatitis. And at the same time, we investigated these two common polymorphisms, as also Miklos mentioned. Hegyi group investigated the same polymorphisms, and they in their German group, and also they did a replication cohort in a French group, and their findings were very similar to ours, namely that they also could not confirm the association between these two common polymorphisms and chronic pancreatitis. So to conclude my first part of the talk, I have to say that common calcium sensing variants do not modify the risk for chronic pancreatitis and should not be considered as genetic risk factors in the clinical settings. And in the next part of my talk, I'm going to talk about the hybrid 1 allele of carboxyl ester lipase. And in 2015, the Anders Moldman group identified a new susceptibility gene, namely the carboxyl ester lipase and its hybrid 1 allele. And this carboxyl ester lipase is secreted in pancreatic acinar cells and also by lactate mammary gland. And it's activated by biliary salts, and its role is to hydrolyze cholesterol, and it helps also in vitamin absorption. And the human carboxyl ester lipase locus is a quite tricky one, because there is this carboxyl ester lipase gene, which has 11 exons, and next to this gene, there is its pseudogene, the CELP, which is highly homologous to this carboxyl ester lipase. And the difference between these two genes is that in the CELP, from exon 2 to exon 7, to the CLP lex exon 2 to exon 7, and in its exon 2 there is a stop codon. And such a constellation predisposed to genomic rearrangements, and such genomic rearrangement is the cell hybrid 1 allele, which is a result of a non-allelic homologous recombination. And I'm not gonna into the detail, but the crossover takes place somewhere in intron 9 between the CL and the CELP genes, and the hybrid allele is formed. And what Anders Moldman groups found was that in their discovered cohort, which contained idiopathic familial chronic pancreatitis patients, they find a very high association of this CEL hybrid 1 allele and chronic pancreatitis. And they did 3 replication cohorts with non-alcoholic chronic pancreatitis patients, and their odds ratio, the cumulative odds ratio of these 3 cohorts is 5.2, and they also could show an association of this hybrid 1 allele of carboxyl ester lipase with alcoholic chronic pancreatitis. So surely it looked that this CEL hybrid 1 allele is a very important risk factor for chronic pancreatitis. And then in the Chinese cohort, they tried to replicate these findings. However, this CEL hybrid 1 allele is not detected in Chinese, Japanese, and Indian cohort. They detected another hybrid allele, the so-called CEL hybrid 2 allele. However, also this hybrid 2 is not associated with chronic pancreatitis. And then a Polish group led by Agnieszka Riegel, they did a replication study in a Polish pediatric patient. And interestingly, there was no significant association between the CEL hybrid 1 allele and chronic pancreatitis in this pediatric cohort. And interestingly, what they found was very high prevalence of this CEL hybrid 1 allele in the control group. So based on this data, we wanted to explore this CEL1 hybrid 1 allele also in the Hungarian cohort. But moreover, in 2020, CASR reported single nucleotide polymorphisms in the CEL1 hybrid 1 allele itself. And they showed that the CT haplotype is exclusively associated with chronic pancreatitis, while the CC haplotype was also present in chronic pancreatitis cases, but also in controls. So in addition to explore the prevalence of CEL1 hybrid 1 allele in the Hungarian cohort, we wanted to see also the haplotype distribution in this population. And so what we did was long-range duplex PCR, and we did also melting curve analysis, and we sequenced all of our CEL hybrid 1 positive samples. And what we found was that we have included 320 patients from chronic pancreatitis patients, and we found in 10 cases the CEL hybrid 1 allele, and among 615 controls, we find this allele only in 5 subjects. So based on our data, this CEL hybrid 1 allele is strongly associated with chronic pancreatitis in the Hungarian cohort, and our results are similar to the original finding by Anders Smolven, in which they reported an OR around 4, and our OR for total chronic pancreatitis is also around 4. And next, we investigated the haplotype distribution of this CEL hybrid 1 allele. And what was very interesting, that all of our positive samples, so the chronic pancreatitis patients and also the controls, had the CC haplotype. So we could not identify the so-called risk haplotype, the CT haplotype, in our cohort. So this needs further investigation, and we are now collaborating with the Polish group, with Agnieszka Riegel, with Hegyi from Munich, and also with Jean-Min Chen from Brest. So we are investigating the haplotype distribution also in these cohorts to find the answer whether there is any risk haplotype of the CEL hybrid 1 allele or not. And so to conclude from this study, we can say that the CEL hybrid 1 allele is significantly associated with chronic pancreatitis in our Hungarian cohort. And with that, I'd like to thank you for your attention.
So Eszter, congratulations. So the question is also to you and for everybody in the genetic field. I might be completely wrong, as Miklos would say in advance, but we have so many negative studies. If I look at the risk factors outside of genetic, like smoking, alcohol, all of them are aggregating each other. Could it be that, for example, okay, like the calcium sensing receptor, there is no association if you investigate below, but would it be to check those where you can see like some alterations, some mutations combined with other, for example, whether if you look at together like the SC26A6 plus calcium signal receptor, maybe you will see odds ratios to be positive, but if you investigate as an univariate mutations, you will see nothing.
Yeah, thank you for your comment, Peter. Yes, it might be that certain risk factors may potentiate itself, but so that should be also hypothesis driven, which one we wanna, which one we think that they might.
I agree, but as Miklos highlighted, these hypothesis driven ones result in completely negative. And I'm more than sure, as Ole also highlighted, that it doesn't mean that you haven't seen an association, that this doesn't affect, and maybe just the methodology, how we try to investigate. If I look at intracellular even calcium, anything, also everything is connected to everything.
So what regarding the calcium sensing, so I'm quite sure that these two polymorphisms do not affect the risk. There might be other calcium sensing receptor variants that are very rare that can affect the risk of chronic pancreatitis. So we have not studied the whole gene, we have not sequenced the whole gene, so that might be that there are certain mutations that might affect the risk.
Yeah, so with regard to the calcium sensing receptor, I think that Dr. Hegyi is exactly correct.
Oh, thank you, David.
The fascinating thing is there's both activating and inactivating. So there's two opposite effects to calcium control inside cells. There are at least 3 areas in which calcium sensing receptors are important. I think Roger Little even showed that that is critical for triggering CCK release in the duodenum. But you can't pick one variant and exclude the use of calcium sensing receptors in clinical practice. In fact, we think more of a burden test where you can begin classifying the activating and inactivating ones, and then you see them commonly with CFTR mutations, but not other mutations, or on the opposite side, alcohol, but not non-alcohol. So the context is important. They're not sufficient to cause disease by themselves. They're not pathogenic, but they certainly are risk factors in the right context. And that's really where we begin to pull the pieces together mechanistically. And because they are, some of them are functional. The ones that you looked at, we would agree are probably benign. But the functional ones in the right context seem to be important in disease. And I think that's how we're beginning to look at it a little bit more mechanistically.
Yes.
So just to Miklos, so the slide that I showed yesterday, when we investigated the utility of calcium sensing receptor gene amongst other genes in predicting the progression from first attack to the different outcomes, we sequenced the whole known variants and the regions of the CASR. It's over 99% coverage. If there's going to be new exomes that we don't know, I mean, we can always modify the test, but there's always coverage for those regions. And that's the beauty of using a test like that. And then when you find a variant and it's classified as pathogenic or likely pathogenic, according to your data, we'll find more of the unknown clinical significance. We go ahead and Sanger confirm it the way you did. When you do it as a panel, you can always remove it if down the line you decide it's not important and throw it out the window. But we can also keep adding the known relevant genes that we are discovering together. That's the beauty of doing things in a panel.
So I think that in research, for sure, we need studies, but in the clinical practice right now, if somebody identifies a very rare variant or even a novel one, I think the clinician, they just don't know what to do with that variant. So you cannot, without a functional study, you cannot classify that variant. So maybe for the clinicians, at this stage, I think it's it's not beneficial to know about the variant which we don't know what it does. For research, for sure, we need to know what kind of variants are in those pediatric patients or in any.
I think if I may comment, we use it also for clinical testing. Not every clinician should interpret this test. You should work as part of a team who has experience in interpretation, who have databases. But the variant-to-variant interaction is what we don't know. And that's where we need to build genetic burden scores and risk scores.
Yeah.
Maybe with the SPINK1 and a certain variant or CFTR, it will be.
Yeah, yeah.
I would like to agree with what she said.
Thank you.
Just a factual question, Eszter, before you leave. So from a kind of pathophysiological point of view, it's a bit along the lines of what David Whitcomb was asking about. So these different mutations, they probably have functional consequences, and the calcium-sensitive receptor in these variants will be different from the normal one. In chronic pancreatitis also, maybe somebody knows data about this, the calcium concentration in the fluids that is in contact with the duct cells may also be quite different, perhaps from in the normal physiological situation. And most of the calcium that enters the pancreatic juice comes from the Eszter cells, which of course have largely died in chronic pancreatitis. So can you kind of guess a little bit in terms of how the calcium-sensitive receptor may have been changed in different situations and how that might relate to changes in the calcium concentration, and therefore can we make some predictions about what kind of consequence they would have?
Oh yeah, thank you very much for this comment. So yeah, so there are these disorders, these familial hypocalciuric hypercalcemia, which we know that there is change in the calcium concentrations, and it might be that these mutations will change the calcium concentration, but then their effect will be by affecting the calcium concentration, such that 336 and or so on. So I cannot comment on the very physiological basis how they may influence the calcium concentration. Our hypothesis was that they might have an effect on the bicarbonate secretion. So upon higher calcium levels, they might stimulate the bicarbonate secretion. So they might prevent from the pancreatic stone formation. So that could be one mechanism how they may prevent from pancreatitis, and certain mutations may lead to disturb these elevated bicarbonate secretion upon higher calcium levels. So that was one of our hypotheses, but it's not proven yet.
So only much of this is speculation still. Even what David said is speculation, because there's no tissue-specific knockout for calcium sensing receptor in the pancreas. So nobody knows what would happen if you remove the receptor. We just don't know.
So, but it would be important to find
I think it would be important to do, because then otherwise we keep speculating back and forth, and the experiment hasn't been done.
I agree with you, Miklos.
Thank you.
I think the other important observation is the original reports of the calcium sensing receptor. There were families that had these calcium sensing receptors and the consequences of hyperparathyroidism and those types of things. Only the ones that also had SPINK mutations got pancreatitis. So it showed a gene-gene interaction from the very beginning. It is not sufficient in itself to cause pancreatitis. That's very clear. But in the context of additional mutations or circumstances, then it contributes and is important for perhaps tipping the balance toward pathologic diseases.
David, this is just conjecture. There's no evidence for this at this point.
No, but that is
it is evidence.
That actually under certain circumstances, something that perhaps in a large population doesn't show up, but in a certain situation, as David points out, it can suddenly become quite critical.
This might be true, but we don't know.
There's different types and levels of evidence.
But that's not an argument for just dismissing it. I mean, it's just an argument that we have to find out what is.
Yes, yes, exactly.
Agreed.
Okay, so before Peter takes over my chair duties, let's move on.